everyone. Now we are going to discuss about poliomyelitis or polio, which is one of the type of water and foodborne diseases. Now the poliomyelitis is a word derived from Greek where the polio means grey and the mylos means marrow or spinal cord. It is also called as, that means the poliomyelitis is also called as polio or infantile paralysis. And this infantile paralysis or the polio or the poliomyelitis is caused by a virus called as poliovirus belonging to the family Picornaviridae, which means pico means small and the genus enterovirus. So as the entero means intestinal related, so it is an infection causing to the intestine. And intestine is going to be related mainly to the food and water. So it's a kind of, or it's a type of food and waterborne diseases. And coming to the carrier or the one which is going to be transmit the disease is only the human. That means the infected patient or the symptomless carrier is going to transmit this disease from one human to the other, that is from one person to the other. So it is going to be exclusively only the human diseases. And then coming to the causative agent, that is about the virus. So here the virus or virion is a spherical particle and measures about the 27 nanometers in diameter. So as it is a spherical, we are going to treat it as an envelope. Okay, and here internally you are going to see a sort of icosahedral symmetry one, which consists of viral proteins like VP1, VP2, VP3 and VP4. And coming to the genome is a single stranded RNA and it's a positive sense RNA. And while the laboratory diagnosis, we can crystallize this virus and can be seen in the cytoplasm of infected cells. And based on the neutralization test, poliovirus strains can be classified into three types. How many types? Three types. Type 1, type 2 and type 3. Okay, coming to the resistance or the tolerance of this virus is, these virion particles are going to be of stable at a pH of 3 to 5. That means they are going to resist our stomach acids. So they can cause the infection very easily by moving to the intestine. But they can be readily inactivated at 55 degrees centigrade for 30 minutes. That means if you properly cook and boil the drinking water, so obviously you are not going to prone this polio virus effect. So the polio virus was stabilized by magnesium chloride against dermal inactivation. So this is all about the causative agent that you are supposed to remember. Okay, so next moving to the epidemiology, as I already told you, this is going to be transmitted with the contaminated food and water. So obviously it occurs by the ingestion of contaminated food and water. And the virus is very stable and can remain infectious for long periods in the food and water. So that's why we are supposed to boil the water and properly cook the food before we are taking in. The virus are going to be stable at pH of the stomach and replicate in the gastrointestinal tract and are going to be excreted in the fecus. So the fecus is the one which is going to be of infecting or causing the contamination of our food and water. And thus these viruses are said to be transmitted by the fecal oral route. And this poliomyelitis is going to occur worldwide. It is not particular to one country or the other country. It is going to occur worldwide. And the disease occurs in all age groups, but children are usually more susceptible because they are going to have the less acquired immunity rather than the adults. Then moving to the replication. So this replication, here you can see this is a virus particle, icosahedral virus particle. And now through the process of endocytosis, this enters into the cell, that the genome only enters into the cell. This code is going to be what we call it as, it is going to be dissolved. Okay, now this whatever the genome is, what is the genome here? It's a single stranded RNA, positive and single stranded RNA is going to occur. Now this RNA The 5' end of RNA is going to have a long sequence that can fold into several stem loops. So that means it is going to form some sort of a loop-like structures. And the VPG protein and the stem loops mimic the cap binding complex, and these permit binding of the poliovirus mRNA to the host cell ribosome. Now the ribosome is going to be that means our ribosome is going to find these looped mRNA of poliovirus, and they start off doing the translation, that is synthesis of the virus proteins, not ours. Now, the proteolytic processing of viral polyprotein will occur. Okay. Now, the virus, as I said, the RNA codes for all the proteins of the virus into a large protein molecule. So, here you can see it's a large protein molecule which is called as a polyprotein. Now, this polyprotein is cleaved into the individual proteins, including the structural as well as replicase, that means the RNA, and the polyvirus. So now the P1 is involved in synthesizing the proteins, and the P2 and the P3, that is viral protein used to synthesize the viral RNA and the structural, they're all going to enter into the vesicle structures and start off synthesizing this replication, and the DNA is going to form, and that DNA is going to synthesize a single stranded RNA, which is nothing but the genome. Now the structural are there, genome are there, and these proteins are there, all together are going to assemble of forming a new poliovirus particle, and now these are going to be released outside the cell by the lysis of this human cell. Okay, so this is all about the replication of the poliovirus. Then coming to the pathogenesis, so already we have discussed that the virus is transmitted by fecal oral route through the ingestion of the contaminated food and water. So once what happens, the mouth, as I said, is going to be a portal of entry of this virus, and now the virus occurs at the site of implantation, mainly at the pharynx region, that is tonsils and our lymph nodes of the neck, and there it enters into the gastrointestinal tract. Now, the virus usually present in the throat and in the stools before the onset of illness. So, that is going to be present in the throat as well as in the stools, because before they are going to show some sort of illness characters. 1 week after the onset, there is a little virus in the throat, but the virus continues to be excreted in the stools for several weeks. Maybe after 1 week, the throat may not have the virus, but the stool, that is a fecus, is going to have the more poliovirus in it for several weeks. Now the virus invades slowly the local lymphoidal tissue and then enters into the blood circulation. There, that condition is called as viremia, and then they will infect the central nervous system, that is our brain, spinal cord, all those things, and where the replication of poliovirus in motor nerves as well as the anterior horn of the brain stem occurs, results in the cell destruction and causing the typical manifestations of poliomyelitis. So these are all the things that are happening, and finally that results in the encephalitis paralysis or the flaccid paralysis. Let's see the clinical features, then you'll get the clear idea. So coming to the clinical features, so we are going to have the three types of polioviruses, type 1, type 2 and type 3. And out of all the things, 95% of the polio cases, the person has no symptoms at all, and those are all going to be called as asymptomatic cases. But the rest of the cases are going to be classified into three types, that is abative polio, non-paralytic polio and paralytic polio. So before these three conditions, we'll have a short thing, that is up to 95% of the cases, as I said you, is not going to have any symptoms, and they are going to be of just defecating the virus in their stool and are able to transmit the virus to the others. So this is asymptomatic cases we say. Then moving to the abative polio, where you are going to have a very minor illnesses like viral symptoms such as fever, fatigue, headache, sore throat, nausea and diarrhea. And these syndromes observed with this form of poliovirus infection are going to be of mainly upper respiratory tract infection, that is sore throat and the fever may also occur. And these all syndromes are distinguishable from other viral illnesses we can find out, and if you are going to have the proper treatment, you can recover within as fast as we can without having the effect of paralysis. Then moving to the non-paralytic polio, so this is going to be of aseptic meningitis, that is symptoms of stiffness of the neck, back and legs, usually following several days after a prodrome similar to that of minor illness, occurring in only about 1 to 2%. Okay, increased or abnormal sensations can also occur, and typically these symptoms will last for, that is sensitivity to light, neck stiffness, all these symptoms will last for only 2 to 10 days, followed by complete recovery. Then moving to the third type, which is going to be called as paralytic poliomyelitis or paralytic polio. Here, less than 2% of polio infections result in falcid paralysis. That means the paralytic symptoms generally begin from 1 to 10 days after prodromal symptoms and progress for 2 to 3 days. And after that, generally no further paralysis occur after the temperature returns to the normal. But the prodrome may be biphasic, especially in children, with initial minor symptoms separated by a 1 to 7 day period from more major symptoms. That is, additional prodromal signs and symptoms can include loss of superficial reflexes, and then some sort of initially increased deep tendon reflexes and severe muscle aches and spasms in the limbs or back. Now, this illness progresses to falcid paralysis with diminished deep tendon reflexes, which reaches a plateau without change for days to weeks. And usually it is asymmetrical, and strength then begins to return. But patients do not experience sensory losses or change in the cognition. But many persons with paralytic poliomyelitis or polio completely, and in most cases, that means they are going to recover mostly, but in most cases muscle function is going to be of what we call it as getting weakened, and paralysis occurs 12 months after the onset, with usually be left with permanent residue. So that's a very bad thing to hear. So now this polio or paralytic polio is again classified into three types depending on the level of involvement. That means how much it is going to affect. So either the spinal polio, that is, it is most common and accounted for 90, that is 79% of paralytic cases, and it is characterized by asymmetric paralysis that most often involves the legs, and that means the whole body is normal, only the legs will be paralyzed. So that is going to be called as spinal polio. Then coming to the second stage is bulbar polio, and this is accounts about 2% of the cases and leads to the weakness and muscles in that is innervated by the cranial nerves or cranial nerves. Then coming to the last one, that is bulbospinal, where a combination of bulbar as well as the spinal, that means all the legs and the things or everything is going to be paralyzed, and this is going to be seen in 19% of the cases. Okay, and even the death to case ratio for paralytic polio is generally 2 to 5% in children, up to 15 to 30% in adults. That is going to differ depending upon the age. So these are all the clinical features where we can see the abortive polio, non-paralytic polio or the paralytic polio. Then how do you diagnose this in the lab? So coming to the laboratory diagnosis, so what are the different samples that can be collected? Or blood, CSF, throat swab, feces, all these things are used to diagnose this polio virus. And if the lab is not too near to the sample collection, obviously we have to use a transport media. So which type of the transport media? That is a viral transport media. Here we are going to use a media called as Hank's balanced salt solution. and that is going to be stored at 4 degrees centigrade for days together, or minus 20 degrees centigrade for months to years. Then how you are going to test it in the lab means there are three methods. One is the viral isolation method, then the serodiagnosis method, then coming to the molecular diagnosis method. Coming to the viral isolation method, poliovirus may be recovered from the stool or the pharynx from a person with pre-assumed what we call it as poliomyelitis. Then isolation of virus from the cerebrospinal fluid is diagnostic, but it is rarely used. If poliovirus is isolated from a particular patient or a person with acute falcid paralysis, it must be tested further. That means we have to go with the serodiagnosis and then with the molecular diagnosis. Then coming to the serology or serodiagnosis, neutralizing antibodies appear early and may be at high levels by the time the patient is hospitalized, and therefore a fourfold rise may not be demonstrated. So we cannot oftenly use this one to diagnose the polio. The next one is going to be of the thing mainly is going to be the cerebrospinal fluid. CSF in poliovirus infection usually contains an increased number of WBC cells, that is white blood cells, and a mildly elevated protein from 4 to 50% mg per ml can be identified in the process of laboratory diagnosis of this polio virus. Then moving to the prophylaxis or prevention and control. So here the prevention and treatment of poliomyelitis is going to be only by vaccination. We don't have any other treatment. So we are going to have the prevention or the treatment is only by the vaccination method. So that's why we are all supposed to have the vaccination at the childhood only. So there are two effective vaccines that are available to prevent this polio virus. One is inactivated or killed vaccine, that is Salk, which is going to be given as IPV, that is in the form of injection. And another one is going to be oral polio vaccine, which is also called as Sabin's vaccine. So here just I want to tell you to avoid the confusion. The Salk vaccine is a killed vaccine. That means the virus was killed here. So Salk, the letter ending is with a letter K, and the killed vaccine means killed K. So here you can remember that Salk vaccine is nothing but a killed polio virus vaccine. So this Salk vaccine is developed by Salk in the year 1954 and contains the formalin inactivated strains grown in monkey kidney cell lines, and it is administered by deep subcutaneous or intramuscular injection. 3 injections are given with intervals of 6 to 8 weeks between the first and second doses, and 4 to 6 months between the second and third doses. The IPV provides long-lasting immunity by stimulating the productions of immunoglobulins called as IgG. These IgG antibodies neutralize the virulent virus that enters the bloodstream and thus prevent the involvement of central nervous system. So this is all about the Salk vaccine. Then moving to the oral vaccine or Sabin's vaccine. So this was developed by Albert Sabin in the year 1962. Okay, and this contains live attenuated strains which are grown in cultures of monkey kidney cells or human diploid cell cultures. The vaccine is administered orally and stimulates both local secretory, that is IgA antibodies in the pharynx, and elementary and humoral IgG. So here we will have only one antibody is getting initiated, that is IgG. But here two types of antibodies are getting provoked, that is IgA as well as IgG. So that's how OPV, that is oral polio vaccine, is going to, or virulent virus, is going to multiply in the cells of the central nervous system, and the virulent virus locks the neurovirulence. So the first dose of this OPV is given along with the DPT at the age of 1.5 months, and the second dose is going to be given, the third and fourth doses are given at the ages of that is second dose at the 2.5 months, and the third dose is going to be given 16 to 24 months respectively. Okay, and it is the OPV which has become the vaccine of choice almost throughout the world. Previously it was used to get, but nowadays we are going to use only the OPV. And this, what we call it as, why we are using this OP means it is highly effective in producing the immunity and very easily getting administered and less expensive than IPV and may spread from recipient to contacts. Okay, so this is all about the what we call it as poliovirus prevention and control. So in this part we had gone through the introduction of the polio. This is a polio virus, and then the structure of the causative agent, all these things, and then epidemiology, how it is going to transmit from one person to the other, that is by fecal oral route transmission. Then the replication of the virus in the cell, then the pathogenesis, how it is going to ingested and how it is getting disseminated, all the things, and then the clinical features, that means what are the symptoms and what are the effects that we are getting because of the infection, and then how you diagnose that in the laboratories, and then what are the prevention and control methods. So this is all about the poliomyelitis or polio or infantile paralysis. Thank you.